Like if it's really unusual, it's like we don't know about the steakhouse. Oh, Cheesecake Factory. I don't know. I feel like that one feels most right in my pocket.
I think an Olive Garden feels the most right, honestly. Or Red Lobster.
Red Lobster one was really funny because they did just a food that had outlets we could play from once. And it was very like endless shrimp.
Okay.
That's awesome.
Okay. So, did you like the surprise on Wednesday?
No.
Why? What didn't you like about it? I love your honesty, but what didn't you like
I
about
wish
it?
there were slides to follow on.
Huh?
You had posted slides.
I didn't.
There was no enzyme slides
Not at first. I see it now, but I didn't see what I was.
on Wednesday, so it was like hard to follow, really. We just free one.
Why? Somebody should have texted me. I was in the building. I could have quickly got on my computer and made it visible. I'm sorry.
I think we just silently suffered.
Huh?
I think we just silently suffered. Yeah.
Don't suffer next time. Email me or have the TA text me. I'm sorry about that. But the surprise was the editor of your book.
Yes.
I mean, that was cool.
Yeah.
Yeah, she's retired now, and yeah, I trained under her. I learned everything I know actually in analysis. So yeah, so I do you remember the topic? Do you remember anything? Well, enzymes. So what do you remember? Any fact about the lecture? Anything at all? Or nothing because you didn't have your slides to go off of? One thing. Thank you. Yes. I'm not gonna ask you what they are, but thank you. That's good.
We have to know.
Do you know what KM is? Yes.
That is the value at the substrate concentration for half of the units.
Wonderful. See, you got something out of that. So that's great. The recording is on Canvas and the slides are now visible. Please review. If you have any questions, please let me know. There will be, I guarantee you, at least one question on the final of the enzymes. Yeah, but we'll have a review session before the final, okay? So we can talk about things together. All right. Let's try to remember a few things about the protein lecture. I'm towards the very end of it. I think I asked that question last time. I will skip the first one. And the second one, we talked about it. What are the 3 reaction steps of Keldol? So,
Digestion, titration.
wonderful. So, when in an exam I ask you about the principle of Keldol, you want to say there are 3 reactions in Keldol: digestion, distillation, and titration. And we follow the nitrogen. Okay, so we follow the nitrogen in each reaction. We calculate total nitrogen and convert to protein. That could be a brief description. It would be nice to know digestion with what? What do we digest with? What was the acid? What was it? H2SO4 concentrated. All right. What do we distill with? What was the base? Yeah, the most common base, NaOH. And then what do we titrate with? HCl, the most common acid. Okay, so if you remember those, you're good to go in terms of principle. Okay. Now, what is the principle of Dumas, which is based? I was going to say the principle. Yes.
Samples combusted. Right.
Yeah.
Nitrogen gas is measured by detector.
Yes, absolutely. So at all samples it's combusted, converts to gases, oxidize like NO2, CO2, H2O, SO2, everything gets scrubbed off except the NO2 or NOx, and they get reduced to nitrogen gas, N2. And the nitrogen gas get detected by a thermal conductivity detector. And then the N is converted, the nitrogen is converted to protein using a nitrogen conversion factor. So what is similar between Keldar and Dunas? Yes.
They both measure protein content by detecting nitrogen.
Very good, absolutely. So both
Both are official methods.
wonderful. Both are official methods. Anything else you want to say about that? Huh?
Isn't it different?
Yes, both give us protein because not all nitrogen comes from protein. Thank you.
I was gonna say crude.
True, yeah. What else anybody was gonna say anything else? They're both very precise if you know at least the titration. Sometimes you have automated titration units as well. You only have to just turn on the machine when it does it for you. So they're both very precise. They both can be close. Good accuracy, they're crude, but they have good accuracy. Perhaps Kelda sometimes have better accuracy than the Dumas because it's based on the immunity, not just the nitrogen. Okay. Very good, very good. So now I'm going to give you a few examples about more research method. We talked about infrared being the method that you do for good quality assessment online during processing, and it just needs to be calibrated by either the Dumas or the Kelda. Very good. Very good, Tim. Okay, now research methods. I'll give you a few examples of research methods. So all the top three are based on reducing power of the protein. So they are all, they have a little bit differences in reagents that we use and method of detection. So I'll talk about BCA, bisynchrononic acid. I'll talk about as an example of an assay based on reducing power. And then a Bradford we did in the lab. So I'm not going to talk about it much. Well, I have a slide on it. And then measuring UV detection. I'll talk about each of those. And we call them research methods because they're not methods you want to go to for a complex food system because of interferences. So for example, BCA, if you have a complex system that has other reducing agents in it, like reducing sugars, you can't use BCA or Duret or Lowry. So it is mostly for protein in solution that you are isolating or you are running enzymatic reaction and you have protein in a clear solution and you want to know how much protein you have in there. So it's mostly research assays that you would use it for. Okay. Well, thought I talked about that. But anyway, so infrared, we have the mid-IR and near-IR. Both can be used for measuring protein in different products. We've learned that IR is based on absorption of vibrational energy, which is the vibration of the bonds. So specific functional groups in the protein, mostly the amide or the amine groups. Very quick. It does require calibration with official methods. Now the BCA. Okay, so it's a mouthful bisynchrononic acid. Can you say that 5 times quickly? No? Okay. Anyway, so BCA is easy to say, and it is based on the reducing power of proteins, especially the specific amino acid residues, cysteine, tryptophan, and tyrosine. Also, peptide bonds can be reducing or have reducing power. So, and the principle is reduction of cupric ion to cuprous ion in the presence of a BCA reagent where you measure the formation of the color. So when cupric is reduced to cuprous, there is a specific change in color that you measure using spectroscopy. And in order to do this measurement, you always have to have a standard. So usually the protein standard, it's not plant protein, but the protein standard is albumin, bovine serum albumin is what is used. Usually it comes with the kit. So BCA is a kit that you buy and it has all the reagents, even the instructions of how to follow the reaction, and it comes with a standard, the bovine serum albumin standard. So we run the standards at different concentrations. We measure absorption at a specific wavelength after a certain time of the reaction, and we get the equation of the line. Then we dilute our sample so that the reading is in the middle of the curve, and we convert the concentration of protein. Again, this method is not necessarily used to detect protein content in a complex food sample. So that you want to remember. It's good for comparative. If you're doing research and you want to compare protein content in different samples, then you can do comparative type of work, but not to really determine accurate amount of protein. The other one, which you did in the lab, is the Bradford, and it's based on protein binding ability to a reagent, in this case, Comasi blue. It binds to the protein, and then also you have a standard. I believe you also use bovine serum albumin as a standard in the lab, and you plot your curve, and you calculate the protein content according to your standard. It is sensitive, meaning you can detect small changes, and it's a easy to do kind of reaction and assay. It's specifically studied not for small peptides because you lose the amphoteric nature of it and the ability to bind to the reagent. And that is another simple form, with HPLC, if we are looking at proteins or looking at trying to quantify or look at relative abundance, the most common way we do is we link it to either PDA or just a regular UV photometer or spectrophotometer. And it's very easy because it's just measuring the absorption at 280, and mostly those 2 out of 3. I mean, you have 3 amino acids, but tyrosine and tryptophan has the highest absorptivity among the 3. So all proteins have some amount of these amino acids. If we want to measure peptides, fractions of proteins or protein hydrolyzing to peptide, normally 280 might not work because peptides, the small sequences, might not have any tyrosine or tryptophan. So we measure at 220, sometimes depending on the reference, 214, anywhere between 214 to 224 is what you see as a range for peptide bonds can absorb at this wavelength. So usually we open 2 channels in PDA or UV variable wavelength detector, and we measure absorption at 220 and 280 to cover our basics. So that's it for protein analysis. Any questions? So next week, you're going to do total carbohydrate assay. So I'm going to start the carbohydrate analysis set of lectures. Well, set of slides. I think we need 2 lectures to finish it. But the first part of it, we're going to talk about total carbohydrate assay. All right. Let me see. Knowledge check. Which of the following is our carbohydrates? Trying to look at Abel. Huh? Well, he says all of the above. Do you agree with all that? Do you all agree that it's all of the above? You're still thinking about it. It's a trick question. Who's gonna say or have another guess? Sophie. Well, it's a good guess. Anybody else would like to guess? B and C, another good guess. Anybody else would like to guess? There's one more answer. You didn't say. It's a lignin is not a carbohydrate. It is a what? It's a dietary fiber, but it's not a carbohydrate. Dietary fiber is anything we consume potentially and our body does not digest. So that's dietary fiber. You know, resistant starch can be that with the dietary fiber because it doesn't get digested. Okay, so pectin and cellulose, because they have a carbohydrate unit in them, the glucose unit in them, or galactouronic acid is the one in pectin, and cellulose is glucose unit, whereas the lignin has no glucose, no carbohydrate. These are the individual lignin subunits, not carbohydrate base. But it is a dietary fiber, it's in plants, and when we measure dietary fiber, it goes with the soluble or insoluble. I'm gonna tell George. No, just kidding. It's in the insoluble fraction of the dietary fiber. I don't expect you to know. Don't worry, I'm teasing. Okay, you're probably working on starch with George, right? Okay, you have excuse. Your excuse. Okay, so yeah, so it is insoluble dietary fiber, but it's not a carbohydrate. Okay. Now, another knowledge check, because this is going to be part of what we're going to talk about in general and total carbohydrate. We're going to talk about total carbohydrate analysis, small carbohydrates, monodi, oligosaccharides. We're going to talk about starch analysis, and we're going to talk about fiber analysis. And part of the starch analysis is not only total starch, we have characteristics for starch that we measure as important in food, which is starch gelatinization. It affects the viscosity in certain food systems. It affects the texture, the dilation. And then starch retrogradation is important, for example, pasta. So you have starch retrogradation that happens within pasta formation. Also, when the bread becomes stale, part of starch retrogradation. But if you want to define what's starch gelatinization, what does it mean to you, that word, when you hear it? What happens to the starch? In simple words. Yes.
The starch takes up water and swells.
Okay. That's all I want to say about that. Yes. So, and in the presence of heat. So you have to have some sort of heat, water, and then the starch picks up the water, swells, gelatinizes. What about retrogradation? What is that process to you? Yes.
It's when the starch granules recrystalline.
Exactly. So starch gelatinizes, and under certain conditions, they reform the aminoplastin recrystallize and cause this. This phenomenon is called retrogradation. And like I said, both are important in foods and both are measured. Okay, so we will talk about those. So, but the question here, can the same method be used for both? Wild guess? Yes. And we will see how one method can be used to measure two opposite characteristics of starch. But we can do that. In the chat. Oh, Rob. Yes, Rob, do you want to speak or do you want to put in a... Oh, I think we just answered the question. Answer the question. Change in viscosity. Yes. So the gelatinization impact. What method? For nitrogen? Okay. Just the percent nitrogen, both Caldal and Dumas gives you the percent nitrogen. And then you use the conversion factor specific to the source of protein to convert the percent nitrogen to percent protein. Ah, okay. So, yes, let's answer this question. This is a very cool question. How do we find the conversion factor? First, what is the conversion factor? Anybody? Oh, Sierra, what were you gonna say?
Based on nature.
Yeah, so what assay do we want to do to get that? Simple, amino acid composition. So yeah, so we do amino acid composition and determine the different types of amino acid, and then determine based on the total amount of amino acid and the different types of amino acid, get the percent nitrogen in that protein source. And then the conversion factor is 100 over that percent nitrogen. The common is 16% is an average percent of nitrogen in foods or proteins. Yes, it excludes nucleic acids. Yes, yes. Because when we measure amino acid composition, we don't get we don't measure nucleic acid, and that's why it's crude, because we assume all the nitrogen that is determined by our assay is coming from protein. So, and we're converting all of that to protein using that conversion factor, and that's why it is crude. Yes, you're welcome. Okay. All right. Good. I think we're going into the next bit. Okay. All right. So why are carbohydrates important? And then we'll answer why we analyze them. So why are carbohydrates important to us? Think nothing, not just food analysis in general. Well, was that your hand up? Yeah, okay, go ahead, Greg.
They let us like do stuff.
They let us do stuff. Absolutely, yeah, so energy, source of energy. Without source of energy, we can't do any stuff. So, very good. Did you have your hand up?
I was gonna kind of say the same thing, but also they interact with other
Yes, so interaction, of course, so like very common reactions are mirror reaction, right? So it impacts flavor, impacts color, so definitely interaction. And then also the interaction with the water gives us
I wanted to add that they also
Function.
functionality.
functionality. Absolutely. What do you want to say?
But it's about the energy. They not only, simple energy, but they're very readily available. And
Yes, readily available energy, the most readily available energy, especially those that we can digest. That gives you the glucose, and that's more readily available than proteins and lipids that have to go through more cycles to get us to the energy. Okay, let's see. And also an important, I'm gonna put it here. Yeah, so you mentioned most of that. A couple of other things like source of dietary fiber. Dietary fiber is important and linked to many health, physiological health benefits. Also, the stability and shelf life. So sugars lower water activity. So you interact with water, reduce the availability of water for chemical reactions. And then another part we'll talk about is also resistant starch. So, for example, if someone is diabetic, you would normally say, okay, if you want to have something rich in carbohydrate because you want something in carbohydrate, maybe eat pasta and less rice. Because pasta has more of resistant starch, graded starch, resists digestion, where rice has more of that gelatinized starch ready to be digested. So resistant starch is important for people with diabetic, and the glycemic index is the rise of glucose in the blood after consumption of certain types of carbohydrate. All right, so therefore, it's important to analyze More specifically, why in terms of food analysis? More specifically, you analyze carbohydrate because,
Part of it.
yes. So on the label, the total carbohydrate is by difference, remember. And then we measure dietary fiber, we measure total sugars, and then this had to go on the label. So compositional information. Also, like everything, quality control and ingredient authentication. So when suppliers give processors their ingredients, it has to come with authentication of certificate of analysis, and often you see carbohydrate analysis there. Research, like anything, of course, so sometimes we need to understand a little bit more the composition of carbohydrates within different foods, how this composition and variation in amylose to amylopectin might impact functionality, stuff like that. And then product development, there's so many sources of starch. Are they all the same? Very different. So cornstarch is different than wheat starch, right? So, or potato starch, yeah. So understanding for product development, what type of starch we need, what type of sugars we need, and so on, what kind of reactions we're after or we want to prevent. Is high fructose corn syrup bad for you?
No,
Why?
all high concentrations of sugar, like anything, too much of anything is bad.
Too much of, yes, too much of anything could be bad, but it's sugar. How is it different than sucrose? What you learned and it isn't digested well as glucose. As you or absorb it as well.
Yeah, I normally just kind of pass it straight through things.
That I don't know, because I thought fructose is small and gets absorbed before it gets to the large intestine. I have to look into that.
The biological, what's the word? It doesn't notify the body that it's full as fast as if you ate sucrose. So
Okay, I'm learning. Good. Yeah, well, you're going deep into the physics.
Fernanda explained it so much better than that.
That's good. Okay, well, what I want to say is that, you know, when you have reducing sugars, you have reducing sugar, whether it's fructose or glucose, they're both reducing. But if you have sucrose, you don't have reducing sugars. So in terms of controlling Maillard reaction, having high fructose corn syrup will potentially cause problems in the heat with Maillard. Well, that was where I was going, but you really took me on a different tangent. But that's good, that's good. We learn. All right. So carbohydrate classification, you studied this obviously in food chem, but very, very simple. Monosaccharides, di and oligo. So oligo, anything 3 or higher and lower than 10 units. And then you have the bulky big ones is polysaccharides, starch and dietary fiber with the exception of lignin. Lignin is not a polysaccharide, so we keep that in mind. Okay. And then, so it is just most of the carbohydrates like starch and cellulose are made up of glucose units. Depending on the disaccharides and the source, lactose is made up of glucose and galactose, and maltose is made up of 2 glucose units, and so on. Sucrose is made up of fructose and glucose. That's it for the intro you have for your food chemistry. Okay, so when we want to analyze a carbohydrate sample, often if it is high in moisture, we start with drying the sample. So that we can have a powder to work with. And if you have high fat, we get the fat out because fat is going to interfere in everything, almost everything. So we remove the water if it's high moisture, and we defat if you have more than 2% fat. You want to get that fat out. In many cases, even if you have a little bit of fat, you just remove the fat. Sometimes you use Soxate, sometimes you just do batch extraction with hexane, for example, or a combination of solvents to remove the fat. Okay, so total carbohydrate assay. So this assay is mostly used for research purposes. So when you have a solution or you have a purified form of carbohydrate and you want to determine the amount of carbohydrate in your matrix, then you can use a simple assay like this one, total carbohydrate. We use it in the lab quite a bit because we work with proteins and starch and maltodextrin, and we want to separate the starches and maltodextrin from proteins. So we want to determine at the end how much of the carbohydrate is left. Can you give me an example when you use this assay, the total carbohydrate assay, phenol sulfuric acid, in your lab? When you use the phenol sulfuric acid method in your lab for research, do you use it? I thought you guys would use it, no? Okay, never mind. I thought maybe not you, the other students of George use it when he's purifying and then he collects a fraction out of chromatography and you want to know how much total carbohydrate is in that fraction. So it's very simple. In foods, you don't usually use it for complex food matrices. In the lab next week, you're going to use it to determine total carbohydrate in simple drinks like 7-Up. And guess what? Beer. Yeah, but not for drinking. So we're going to measure total carbohydrate in drinks because it's simple. They're simple matrices and you're able to get, there's no dietary fiber, for example, you're able to convert everything into glucose and get total calories calculation. Okay, so what is the phenol sulfuric acid method? So this is a method that you basically have your drink sample and you add to it concentrated H2SO4. As soon as you add the H2SO4 to your sample, it's an exothermic reaction. Heat is going to be generated. And that heat and acid is going to break everything into monomers. Whatever you have in there. If you have starch, it's going to break into glucose. If you have sucrose, it's going to break into glucose and fructose. So basically, you convert everything into monomers. And these monomers, the monosaccharides, also in the presence of acid and heat, will convert to furans, which is an enolization dehydration. Enolization is production of double bond, and also dehydration is removal of H2, water. So you get a mixture of furans. Depending on what monosaccharides you have, you get different types of furans, and that furan, then you add phenol substance to your reaction, and that phenol is going to react with your furan to give you a colored product that you will measure using spectrophotometry. Very simple, very easy, but the most dangerous lab, according to Cindy, because you're working with really, really concentrated acid. So you're there under the hood dispensing H2SO4 into a tube that has your sample in, or your standard. And the reaction is very quick. And so the speed and how you dispense the H2SO4 will impact. So if you don't do it really the same way for each tube, you'll get really poor precision. So when you hold that cube and use the reed pipette to dispense the H2SO4, make sure you use the same speed. And then right in the middle of the cube, not on the side, so it all goes into the sample. That will get you the best precision. Okay, so usually we use a glucose standard. So for that assay in the lab, you will be using glucose and you will prepare your standard at different concentrations. And the standards will go through the same process, adding the H2SO4, and then you add the phenol later. And you measure the change in color, and the lambda max, or the absorbance at which, or the wavelength at which you have the highest absorbance, is somewhere between 480 to 490. So you would choose one wavelength and you will measure and plot the standard curve and get the total carbohydrate. Now, what I have here, the reaction is non-stoichiometric, meaning it's not, the quantification is not 1-to-1 always. It depends on the furan, the chemical structure of the furan. So even if you have 1 mole of glucose and you have 1 mole of fructose, they won't give you the same intensity of the color. Okay? Even if it's 1 mole and 1 mole, fructose will give you a furan different than the glucose furan, so that absorbance will be different. So the standard we use is usually impacts the accuracy of the results. So if I know that the composition of my sample is a mixture of glucose and fructose, then I try to use a standard that has both in it, so that I'm more accurate. However, if you're using it as a comparative across a set of 10 samples, and you want to see how different they are from each other, it's okay to use glucose as a standard. All right. So basically, in one sentence, the accuracy of the assay depends on the different monosaccharides you have in your sample and the standard you use along with it. Questions? So this is what you're doing in the lab, very simple assay, and you're going to do a bunch of dilutions and run the assay and do calculations. In 7-Up, in 7-Up Light, in beer, and in beer light. So these are the samples for next. All right. Any questions about that assay? All right. Yes, sure, Rachel, what do you want me to? Yeah, the accuracy of the total carbohydrate assay here depends on the different monosaccharides that are in your sample, or when you hydrolyze them, if you have starch, or if you have sucrose. When you hydrolyze that, gives you a monomer. So the accuracy depends on the monomers produced, the furans that come from these monomers, and accordingly the standard value. If you know that you have fructose in your sample, it might be a good idea to have fructose in your standard. Otherwise, if you just use glucose, it won't be as accurate. Yeah, because it is not a stoichiometric reaction. That means 1 mole of glucose will give you a different reading than 1 mole of fructose. Okay. Yes. Yeah, we're using a glucose standard in the lab. Yeah. They're different. Well, so in what do they use in Sprite, regular Sprite? Do they use high fructose corn syrup, or do they use sucrose? Yeah, that's a good point. We have been using it with glucose because we also use the same standard to determine alcohol content, well, not alcohol, sugar content in the beers. So, and where the sugar content in beer usually mostly from, yeah, when it breaks down the alcohol, whatever it is, right? It breaks down the glucose. So we use that. Okay. Good idea for next time to have a standard glucose and standard glucose and what happens. Good thought. Keep it in mind. I hope I remember next year. Okay. All right. All right. So the rest of this is broken down into analyses of mono, di, and oligosaccharides, so the small carbohydrates. And then we're going to talk about analysis of starch and then dietary fiber. So it's broken into three components. So the first component is determining mono, di, and oligosaccharides in your sample. So we can do that using different methods. Colorimetric methods, we have a few of them, and all three listed here are based on measuring the reducing power of your monomer, or mono, di, or oligosaccharides, assuming that all of them will have a carbonyl carbon reducing. And I'll tell you what we do with sucrose. Sucrose is not doesn't have a free carbonyl. So all of these three methods are based on the reducing power, and in all of them, basically, cupric is reduced to cuprous. Now, the most common is Smoogie-Nelson. All these names are what? Based on what? They have this habit of calling assays by the name of people that developed them. So these are folks, guys. I don't know if guys or ladies, but folks. All right, so Smoogie-Nelson is the most common. The way they differ from each other is the reagents that we use in the assay, whether it's based on titration or spectrophotometry or gravimetric by weight, measuring weight. So, and then the chromatographic method, we'll talk about chromatography using paper and thin layer chromatography or using HPLC and GC. So here's an application for using chromatography that we studied earlier. Or we use enzymatic, and they are very, very common. Especially most common is the glucose enzymatic assay that we use not only to measure just total glucose, but it's used to measure starch, total starch, after breaking all the starch into glucose monomers. So these are the methods. We're going to talk about each one of them, each set of them. So, but first, if you have a complex system where you have starches, dietary fiber, protein, and some of these sugars and oligosaccharides, you want to get those small molecular weight saccharides out, separate them from everything else. So that's the first step. So after you dehydrate your sample and defat it, remember, whenever I ask you a question in a quiz or an exam, what are the first steps that you follow for total carbohydrate analysis? Well, define moisture, remove moisture, and then remove fat. Is that right? And then we start the process. And the process is get those mono, di, oligosaccharides out of away from your starch, protein, and fiber. So a very common extraction that is efficient is using 80% ethanol. They are soluble in water and they are soluble in 80% ethanol. We prefer to use 80% ethanol because it's easier to evaporate and get rid of it later on. So what you see here, so you weigh your sample in a little, not little, a round bottom flask, and you have it in a water bath, and you heat it up, and you will put your 80% ethanol, you heat it up, and you put it in a kind of a reflux system where you have a distillation. So you tap water can go in and out just as a cooling. So what happens is your ethanol will evaporate, but it's also solubilizing your protein. Gosh, I love proteins. It keeps coming up. You solubilize your sugars, but you want that heat to help it and get all your sugars out, but you don't want to evaporate your solvent. So you put that reflux system to, once the solvent evaporates, it cools down and comes back into the flask, so you don't lose it. After that, you separate your solids from your 80% ethanol that has your sugars in it, and then you use a rotovap. You all saw a rotovap in the lab for the isoflavone. So basically, it's evaporation of the 80% ethanol under vacuum. And using distillation, you collect the solvent here, and then your sugars remain in that original flask. And then you can solubilize it in water, in whatever is your the agent that you solubilize it in for measure. So that is how you extract it. Another way, which is not very common, is called the Caras treatment. So you put reagents specifically that will precipitate proteins and large carbohydrate and colors, and then in the solution, your sugars remain. Okay. So I have a couple minutes to go over the principle of the reducing sugar assay, the colorimetric. So once you got your sugars from your matrix, then you can determine reducing sugars, total reducing sugars, using cupric ion and a reagent. The reducing sugars will convert cupric to cuprous. The cuprous react with the reagent, and a color is generated in simple form. So you can do gravimetric because you get cuprous oxide precipitate, a red precipitate. So you can actually weigh it and convert that weight to the original amount of reducing sugar. Of course, you need your standards. And then, oh, I forgot I have that. This is just to remind us that you really need to dilute the sample. If you have a lot of sugar, then you get a lot of reaction, then it's really hard to quantitate. So the three different methods, what I want you to remember, all three of them are based on the reducing power and reducing cupric to cuprous. They use different reagents, and you can do titration, gravimetric, or spectrophotometric, photometric, depending on the reagents that you use. That's it. I don't want you to remember the names of the reagent, but it's just based on reducing power of the sugar. Lastly, this is really quick, you guys. If you have sucrose, what do? You heat it up in acid, break the bond, glycosidic bond, then you have glucose and fructose, then you can apply the same method that you choose. And that's it. That's good. I'll see you on Monday. Have a great weekend. Yes, good question. Charu Dunn, she is really speedy in grading. She gave it to me last Friday. I didn't want to give it back to you without me looking at it, so I wasn't able to. I will do this weekend. I promise I'll bring it. Great, thank you. Yeah.
And what about the quiz next week?
What about the quiz next week? What about it? What's your question? We have a quiz on Friday. What was the last thing you studied? I think it's on that IR, yeah. So all of the properties and
Okay.
I will confirm. I don't think, I'm not sure if Dr. Fabare will be on it. Let me look at my notes, but I will confirm.
Thank you.
Yeah.
You have a wonderful week.
Yes, thank you, you too.
Yeah. I'm fine.
I think.